Bismillahirrahmanirrahim. Assalamu alaikum friends. Today we will discuss about rapid amplification of cDNA ends. So stand by a race mean that a rapid amplification of cDNA ends. Now let's go to start that why we do a race technique. So basically race used for to determine the unknown sequence of messenger RNA that are basically the 3' end and the 5' end to determine these sequence. So we use arrays technique, rapid amplification of cDNA ends. So arrays have two types. The first one has race 5' end, and the another type is race 3 end. So we used two types of race mechanism to determine our messenger RNA planking region. So race pipe are used to determine the pi' end of the messenger RNA, and race 3 are used to determine and to known the 3' end of the messenger RNA. So let's go to start the race pipe technique, how we can determine the pipe prime end of messenger RNA. So let's have some steps. Now let's go to start the race 3. Sorry, race 5. Now this is our messenger RNA, and here the 5' and 3' are present. Now, in first step, we convert this messenger RNA into a cDNA. So for that purpose, we design a primer in this region. So this region are already known, but the flanking region is unknown. So we first design here a random primer to amplify this region. Now we used primer and reverse transcriptase enzyme. So the reverse transcriptase enzyme work to transfer this RNA into cDNA. They add nucleotide and they make the cDNA. Now, when we add the primer and RT enzyme and a PCR tube, so what happened? This primer are attached to this sequence. Now this is our known sequence, and here the reverse transcriptase RT enzyme work and add nucleotide AT, for example ATC, GA, and so on. And this is the 3' and 5'. Now when the cDNA are formed, this is our complementary DNA. So we use another type of enzyme that is basically called RNase H enzyme. So RNase H enzyme work to degrade this extra DNA, sorry, this messenger RNA. They remove the messenger RNA from this cDNA. So when we use this RNase H enzyme, so automatically this messenger RNA will be removed. So we have uh, produced the cDNA. This is our cDNA, and here the 3' and here the pi prime are present. Now after the formation of complementary cDNA, we used another enzyme to know this to determine this 3 prime sequence. So we here we use deoxynucleotidal transferase. So, deoxynucleotidal transferase aid poly A tail to this 3 prime end. So, when we add this deoxynucleotidal transferase, so some modification are takes place in this 3 prime end. So, here the poly A will be attached. Now, in next step, we prepare a poly T primer which are complementary to this region. So we add poly T primer and another material which are basically take polymerase. So when we add the T primer and take polymerase, so uh, here the poly A are present. And when we add the poly T primer, so they attach to this region. And here take polymerase start its amplification. Now we have produced a double stranded DNA molecule. After that, when we make this double stranded DNA molecule, we do multiple time PCR for this double stranded DNA amplification. So we make a large copies number. Now after making a large copy number, we do sequencing. We do sequencing for this region, that what region are come here. So this is all about the race pipe race process. So we determine the pipe prime end. Now let's go to start the 3 prime race. As compared to pipe rays, pipe prime rays, the 3 prime rays are very easily processed. So this is the our messenger RNA, and here 5 prime and 3 prime. Now we know that in 3 primer the poly A tail are present. So we directly make poly T primer for this type for this region. So we already know that here the poly A tail are present. So we easily make poly T primer for this sequence. Now we add poly T primer plus RT enzyme, reverse transcriptase enzyme. So what happened? This is our 5 prime and this is 3 prime. Now when we add poly T primer, so this primer attached to this sequence T T, T, and the RT amplify this region and make this complementary DNA. So this extra region are unwanted. So we remove this extra region by using RNase H enzyme. So when we use this enzyme, so we obtain the complementary DNA 5 prime. So this is our complementary DNA are uh, made. Now after obtain this complementary DNA, so what we'll have to do, we use the primer. We use another type of primer which are poly, poly A primer, and another enzyme which are take polymerase. So this poly A, this poly A, sorry, this is T and Cdna. This poly A primer attached to this region. This is 5 sequence, and take polymerase, add nucleotides to this new region and complementary region. After doing this work, we make multiple multiple cDNA copies. After large copy number production of cDNA, we do sequencing. So after sequencing our unknown 3 prime will be obtained. So this is all about rapid amplification of cDNA, that how we can produce complementary DNA, and how we can determine the unknown sequence like 3 prime sequence and a 5 prime sequence. So hopefully you understand this lecture. If you are new in this channel, so please like, subscribe, and share this link to our friend. So see you in next time.